excited to have Dr. Alana Church here with us today. Everyone, welcome to OsteoBites. My name is Christina Iptoma, and I am mom to OsteoAngel Dylan and the Director of Scientific Programs at MIB Agents. And today on OsteoBites, we're talking with Dr. Alana Church from Boston Children's Hospital about the study that was recently published in Nature Medicine just a couple weeks ago with Dr. Katie Janeway and many other collaborators demonstrating the clinical utility of molecular tumor profiling for children with cancer. Thanks so much, Dr. Church, for joining us on OsteoBites today. We are so thrilled to have you. This paper was just published recently, so we're really excited to get the scoop and this really timely update from Dr. Church. And thanks to Vicki for joining us as our panelist today. Vicki is an osteo warrior, and she's also on our MIB Agents Junior Advisory Board. So first, a little bit more about our guest today. Dr. Alana Church is a board-certified pediatric pathologist and molecular genetic pathologist. She's the Associate Director of the Laboratory for Molecular Pediatric Pathology at Boston Children's Hospital, and she's also an Assistant Professor at Harvard Medical School. She's participated in several high-impact studies using molecular profiling for pediatric cancers, including the ICAT study, Profile study, and the ongoing multi-institutional GAIN Consortium study. Her career is dedicated to implementing high-quality, accessible molecular profiling tests to support the care of children with cancer. And before we get started, I just have a few announcements. We are back from Factor, which was amazing. We took a couple weeks off during Factor, but here's what you missed if you weren't there. The Osteo Warriors defeated the Docs in our Docs versus Warriors opening ceremony and took back the Golden Bone. And Vicki here beat Dr. John Healy, who's like legendary orthopedic surgeon from Memorial Sloan Kettering. She beat him at the game of operations. So I think, Vicki, you have a new career to explore. We had lots of collaboration with our pre-Factor mingle at Mike Hess Brewing, our 8 scientific panels with 50 speakers. We had wellness sessions and seminars for patients and families. And we even had a Healing Hearts session for bereaved families. So there was really something for everyone this year in the osteosarcoma community. And more importantly, it brought us all together to collaborate for a cure. So we'll be sharing more highlights, videos, and photos soon. Keep an eye out for those. And we have even more events coming up this summer. July, as you all know, is Sarcoma Awareness Month. So please join us for our virtual Outrunning Osteosarcoma event, where you can run, walk, or cycle with us, and you can help cause a cure. With MIB Agents, no one walks through osteosarcoma alone. So let's go out running together, and we'll put the registration link in the chat. Also, to commemorate Sarcoma Awareness Month, MIB Agents is partnering with Kendra Scott for a multi-city give back event taking place the weekend of July 15th to the 17th. By making a purchase during this weekend, you are helping raise funds and awareness for osteosarcoma. There's also a promo code that you can use online, and I'll put that in the chat as well. And if you aren't familiar with Kendra Scott, definitely Google her, check it out. She makes really great jewelry. And then in September, we encourage you to be bold and go gold for Childhood Cancer Awareness Month. You can make and sell bows in your community or make a donation this summer and get a bow that shows you care. And you can contact Anita, our events manager, to volunteer, and I'll also put her email in the chat. And we are very grateful to the Osteosarcoma Institute. This episode is sponsored by the Osteosarcoma Institute, OSI, which is a nonprofit organization led by osteosarcoma experts from top US cancer centers who together are concentrating on the cure for osteosarcoma. The mission of OSI is to dramatically increase treatment options and survival rates in osteosarcoma patients through identifying and funding the most promising and breakthrough osteosarcoma clinical trials in science. In addition to advancing research, OSI also provides a free resource called OSI Connect for osteosarcoma patients. Our osteosarcoma experts can discuss available treatments, possible side effects, and provide helpful advice for getting the most out of your visits with your treating physician. And this resource is available in English and Spanish and aims to help patients and families to find answers to their questions. All right, so thank you, OSI. And I'm going to hand it over to Vicki to introduce herself.
Hi, everyone. My name is Vicki. I think Christina already did a good job of briefly introducing me, but I am a Junior Advisory Board member, and I was so happy to be at Factor not too long ago. And still really surprised that I beat Dr. Healy at operation, but we had to do it with our non-dominant hand. So I guess I'm just a little better with my right hand than his left hand, but yeah, that's kind of about me.
All right, great. And Dr. Church, it already looks like you got your slides up, which is great. We'll just hand it over to you to get started.
Great. Thank you both so much. It's so great to be here. And I also had a great time at the Factor meeting. So it was really fun to see everybody there. And I'm really pleased to be here with you today. So I get to be the one to talk about our paper that just came out a couple of weeks ago, Molecular Profiling Identifies Targeted Therapy Opportunities in Pediatric Solid Cancers. So just an overview of what we're going to talk about today. What did we do in the study and why? What did we find out? And what does it mean for the sort of greater community? So I get to be the kind of mouthpiece of this project, but there's a ton of authors, as you can see, all these names on the paper. So it's very, this is the work of many people over many years to bring this to you. And I'm happy to be able to speak on their behalf, but I don't take credit, in any way for all of the work that's gone into this. So the GAIN Consortium study is a prospective cohort study enrolling patients aged up to 30 years with advanced solid tumors. And we enroll patients from 12 children's cancer centers all across the United States. So the format of the study is that we enroll oncologists, patients, and families, and they are consented to the study. And then we take tumor samples from their tumor from any procedures that they've had in the course of their clinical care, whether it's a biopsy or a surgery. And we do some testing using next generation sequencing as sort of the core testing. So it's a molecular tumor profiling assay that we're using and identifying alterations that are really aimed to influence the care of the patient. So the primary aim of the study is to identify molecular alterations, which might give us another option for a targeted therapy for the patient. Every patient who has a test that goes through this panel gets a report that goes back to their oncologist explaining the results and some of the ways that that might influence their care. There's a ton of work that happens in the back end on the data collection because there are so many patients in this study. We have over 800 patients enrolled now, keeping track of all of the patients and all their information, the sequencing results, and then all of the evidence and reports that are going back to them.
Dr. So you're freezing up a little bit.
Oh dear. I'm sorry. I'm not sure what to do. Should I go back and did I miss anything important? I'm not getting a message about my internet connection. I'll just try to forge ahead, but please tell me if you've missed something.
Yeah, I'm just seeing Christina kind of break in and out. I think your Wi-Fi is okay though.
Oh, okay. All right. Well, let's go ahead and please, Vicki and Christina, tell me if I should back up. So I'll just step away from the study for a moment just to talk about what we mean when we say molecular alterations and molecular profiling. So a molecular or genetic alteration is a change that happens in the DNA within the cancer cell itself. And so this is a difference of the DNA between what's going on in the cancer cell and what's happening in the other cells of the body. And these genetic changes are really what drive the tumor to grow and, to be the type of tumor that it is. And there are also changes that can happen in the DNA which might influence the care. So these alterations can give us some clues about how, about a specific diagnosis. There may be some genetic alterations that influence the prognosis. So they give us a hint that the tumor may be more or less aggressive than others that look the same under the microscope. And some molecular alterations are associated with response to targeted treatments. And so the targeted treatment landscape really has changed a lot over time. So new drugs are being developed all the time, and pairing the molecular alteration with the available drugs that are available now, is an ongoing project and a lot of work. And so that's a lot of the work that's happened behind the scenes in this study as well.
And Alana, does it also cover RNA, changes in RNA when you talk about changes in the DNA?
Yeah, so the way that those they're related. So if we think about sort of tumor biology or any sort of genetic biology, the DNA is kind of the blueprint for the cell and what the cell is going to do and all the proteins that's going to make. And then the RNA is like the in-between step. So the DNA makes RNA, RNA makes protein, and then the proteins are the ones that kind of like do the job of the cell. And the RNA is present in the cell in sort of a transient way. And we make use of both DNA and RNA sequencing assays. So sometimes it makes more sense to go look at the DNA as sort of the primary target, and sometimes it makes more sense to look at the RNA. We'll talk in a few minutes about gene fusions, and the gene fusion assay that we primarily used in this project was based on RNA. And for many reasons, mostly because we get rid of some of the introns, which are really long sequences in DNA that are difficult to assess. The RNA is really a nice way to assess for gene fusion. So we have a combination of DNA next generation sequencing and RNA next generation sequencing. And on the technical side, I think some of these results will sort of advocate for using that RNA sequencing, especially when we're looking for gene fusions, which we'll get to in a couple of minutes. That was a very sophisticated question, Christina. So if we want to talk about the cancer genome and what do we mean by that? The model that we typically think of, or that most people think of if they have thought about, cancer genetics, is this one that really models what an adult carcinoma looks like. So the more common cancers that we see in adults, like breast cancer, lung cancer, colon cancer, follow this kind of progression where, you start with the genetics of the person, before they had any cancers, and then the cells acquire mutations over time. And at some point, the cells have required so many mutations of just the sort of right or wrong flavor, depending on kind of how you think of it, to turn that cell into a cancer cell. But the pediatric cancer genome really works quite differently. A lot of the time, there's not an accumulation of mutations over time because the child hasn't lived for all those years, right, to have even gotten to that point. But they often have a single, genetic event that's more like, I put a lightning bolt here, but one big change that really switches that cell from being a non-cancer cell to being a cancer cell. And then everything kind of flows from there. So we have to think differently about how we test and what different types of alterations we're looking for when we're thinking about a child with cancer, because they really don't have the same cancer genome that an adult with cancer would have. And so many of these events that we see in this study, and others have shown this in studies of pediatric cancers as well, are gene fusions. So when we talk about gene fusions, there are two genes that usually live far apart from one another, within the cell, but on different chromosomes, for example. And there's a break that happens in both genes, and then the parts of the two genes come together. So you have sort of half of one and half of the other, and they create this hybrid gene sort of at the DNA level, which also gets translated into RNA and then creates this hybrid protein. And that can be a very powerful driver of cancer. So the example that I have here is a TP53 USP6 fusion, which is one that we actually saw in one of the patients in this study. And I'll describe why that was important for that patient. But we have seen fusions over and over again in the pediatric cancer population. And we really, show here how important the gene fusions are when we're thinking about those clinically relevant alterations that can impact their diagnosis and their treatment options. So the reason that we did this study and sort of set it up the way that we did is that molecular tumor profiling, using these kind of newer techniques like next generation sequencing, have really become more the standard of care for many adult cancers, but still is not routine for many children with cancer, and including solid tumors. So a great example on the adult cancer side is lung cancer. So just over the last 10 years, a patient now who has lung cancer would be, the standard of care to have many genetic tests done to identify different molecular alterations, which can really impact their therapy. And then the therapeutic decision-making, really flows from the results of those studies. And the vision behind all of this really, is all thanks to Dr. Janeway, who is the senior author of this study, and knew, many years ago that this is the path that we would be on, and that these data would really be needed to demonstrate the importance of doing these assays, not just on a research basis, but really for clinical care for children at the time of their diagnosis to really direct their care going forward. So these are some of the conclusions that we found from the study, and we'll walk through some of them in more detail. But most importantly, clinically relevant molecular alterations were identified in most of our patients, 86%. So the molecular alterations are shown to be important for selecting targeted treatment. They have an important impact on the diagnosis. And gene fusions are really important, and they're common in this patient population. So let's start with the treatment. Oh, I'm going to progress my slide here. Okay. So we'll start by talking about the treatments, but first let's just talk about who's in this study. So the enrollment criteria are very broad. So any child with a solid tumor, extracranial solid tumor, who is either newly diagnosed, relapsed refractory, but really at a high risk, or they have an uncertain diagnosis, was eligible. So we have a lot of different diagnoses represented within this cohort. This cohort represents the first 345 patients who had successful sequencing done through the study. And this mix of patients, I think, really reflects what a lot of pediatric oncologists and pediatric pathologists see in their practice, which is that there are a lot of rare tumor types. We have a lot of sarcomas. So osteosarcoma, as you can see, is our most common single diagnosis. And then if we put all the sarcomas together, represents more than half of the cohort. And then a lot of rare tumors, even that other category, even though we're already into the rare stuff, the other category is quite big as well. And again, this is some of the work that's going on in the back end. When we're talking about therapeutic alterations, we have a huge group of people, because we have participants from all of these different cancer centers, really thinking through every molecular alteration that we identified and the potential for it to be a therapeutic impact for the patient. So we keep track of all of those. We have these tumor boards, very often, where we discuss new trials that are coming up, complicated cases, and then the reports get generated from that. And I think also importantly, for this study, when we were doing the analysis, we kept track of the reports that went back to patients at the time that their assays were done, and those reports went back in real time. But we also, as part of the study, reanalyzed every variant so that we were doing them all at the same time, because there are changes over time. Even an alteration that we might have seen a couple of years ago, might point to a potential went on a clinical, and maybe the trial closed now. And I knew there were resolutions over time, which I think also sort of speaks to some of why this can be difficult for people to kind of do in real life, because it's so complex. But we have a great team, so that was great for us. So if we're putting all of these together, so for every patient, we're identifying alterations that are potentially relevant to their therapy, their diagnosis, or their prognosis. And here we have our 345 patients, and each little figure represents 10. So you can see that most of the patients in the study had one or more alterations that would point to a potential for a therapy, prognostic alteration, diagnostic alteration, or a combination of those, and a very much smaller number that really didn't have anything that we were able to find to potentially impact their care. So when we're talking about matched targeted therapies, what we mean when we say that is that there's a specific molecular alteration that we found, through one of these sequencing assays that points to a specific drug that would target either that alteration or the pathway that alteration is influencing. So just to kind of clarify, because there's a lot of different ways to think about treatments. This is a very complicated diagram that was not made by me, but Hannah Como, who's one of our wonderful research coordinators created this graph. So what we're looking at here is a way of sort of representing some of the many therapeutic recommendations that were made. So on the bottom, you can see the gene. And so if we look at, for example, the left side, we can see TP53 is a gene there. And if we look up and down, we're looking at different drug classes. The size of the dot represents the number of recommendations that were made. And there are different drug classes that can be recommended for different genes. Some of them, are in the same pathways. And then there are different trials that sort of open and close over time. So for any one gene, you may have multiple dots on that line, and so on. So this is really, again, representing some of the complexity here.
Yeah, this is an incredible chart. And Alana, for just to clarify, so for all the sequencing that was done, this was using the OncoPanel?
Yeah.
Okay.
Yeah, so every patient in this cohort had OncoPanel sequencing done. That's sort of the core technology for this study. And all of those 345 patients had tumor tissue that was available, and they were able to sort of successfully get a report out because the test worked. We had enough DNA to make the test work. We also did RNA sequencing on many of the patients, and we show in the paper in sort of the supplemental figures the criteria that we would use for whether a patient does or doesn't get RNA sequencing. And it really depends on the diagnosis. So if we found everything that we were looking for, in the OncoPanel assay, then they sort of, typically stop there. But for a lot of the sarcomas or tumors that are really driven by fusions, we added that RNA panel because that really helped us to get more information about the fusions. All right, so when we're thinking about treatment recommendations, so we had 200 patients who had an ICAT, which was the tiering system that we use to assess treatment evidence for match targeted therapy. So 200 patients had an ICAT recommendation and were alive at the time the results were received. And then we followed them through to see what happened and what kind of therapy they sort of pursued. So we have both patients in the newly diagnosed category and also patients who were in the relapsed and refractory category. So many of them went on to get unmatched treatment, and the study itself was not directing anybody to do or not do, any treatment. Everybody was making their own decisions, with their oncologist about what was right for them. And so many of the patients, again, depending on their stage and what was happening with them, were following really more standard of care. So they were getting unmatched treatment. Some of them weren't getting treatment at all. Maybe they had been just treated with surgery. And some patients did go on to get matched treatment. I think it's also notable on these graphs that you can see that some patients had ICAT recommendations made about treatments that they could pursue, but that the targeted treatments actually weren't available to them. So that's another role for advocacy, for us going forward.
Well, I was going to say, and you might be getting into this in your future slides, but just what some of those barriers were or reasons for not pursuing the matched treatment. So you mentioned, like, in some cases there wasn't therapy available, or in some cases insurance doesn't cover the medication, so the patient can't pursue it because it costs like $20,000 a month or something.
Yeah, I don't have that granular of data about what happened for those 20 patients and why they didn't, end up pursuing or sort of having those treatments available to them. But I expect, as you just mentioned, that there's really a variety of reasons. I mean, some of the things are geographic too, right? If there's a clinical trial available, but it's not anywhere that the patient could actually get to, is it really available to them? So there's a lot of complexity, as in sort of pursuing these, newer treatments and things that are still on trial. I mean, one of the other things too that often comes up is the age of the patient. Sometimes there's a drug that's available for adults, and that may not be available or may not have the safety, profiling done to make it accessible to younger children. So that's another, potential barrier out there.
Great. And I was just going to say, Dr. Janeway's in the chat right now.
Oh, good.
And so she said she could comment. So I asked her to comment in the chat because I'm not quite sure how to get her on screen on the webinar. So
Yes,
Thanks,
she's
Dr.
she's the one that holds all of those details for us. Okay, so to give an example of a patient who did get treatment, match targeted therapy, and was one of the ones that we would, classify it as a responder. This patient is a 14-year-old boy who had a grade 3 neuroendocrine carcinoma of large cell type, and we found through the study a gene fusion. So we'll keep talking about those gene fusions. So this is a BRAF fusion, and BRAF is a gene that has fusions and alterations, and also is one that potentially is targetable by a match targeted therapy. And so this patient did get a targeted therapy of trametinib and had a partial response, which lasted for 5 months. So this was one of our 7 patients who we counted as responders to match targeted therapy. If we look at all of the patients together who had who were treated with match targeted therapy, you can see them all here. This is what we call a swimmer's plot. So we group them together. The top group is the ones who responded. So we have our 7 patients. The patient that we just saw on the prior slide with neuroendocrine carcinoma is right here in the middle, was treated with trametinib, and then we can see how long the treatment lasted for and what was the best response. And so as you can see, sort of going through this, we have a small group of responders here. We have patients who were not eligible to be a responder, and then some patients who got match targeted therapy and progressed on therapy. And I'm glad that Dr. Janeway is here, because if there are complicated questions about this graph, she's really the best person to answer those as well. I think one of the things that I really want to point out that it's important, of these 7 patients who were responders, 6 of the 7 responded to drugs that match to a fusion. So of the top 6 here, you can see there's a Notch1 and NTRK fusion, 2 BRAF fusions, RET fusion, and ALK fusion. And then there was another patient who responded to a sequence change, a therapy that matched to a sequence change. So again, these fusions are sort of showing themselves to be very important.
Sorry, could you just explain the not eligible to be a responder with that?
I think this is a question for Katie too. So yeah, I think I can't recall. I'm sorry. Let's ask Katie when we get to the chat.
We'll put that in, Dr. Jane, if you can put that in the chat, that'd be great. Thanks.
Okay, so if we think about the impact on diagnosis, although this wasn't really a primary aim of the study to identify diagnostic alterations, this sort of comes up, and this is certainly in my wheelhouse as a pathologist, because we think about diagnosis all the time, and pathologists are really using a lot of these sequencing assays at the time of diagnosis to support or to help us make a specific diagnosis. So here we have another patient who's an example of a patient who had a real-time impact on their diagnosis. So this young man presented with pain in his leg and had a tumor that was identified in the bone on imaging that you can see on the left. Was treated at first, with sort of more conservatively, and had a diagnosis made of an aneurysmal bone cyst, which is a benign tumor. And then the tumor recurred, and so was sort of not quite behaving, in the way that we might expect an aneurysmal bone cyst to behave. Had another pathology assessment, and looking under the microscope, the cells looked more aggressive than you would expect to see, so more pleomorphic than we would expect to see for an aneurysmal bone cyst. And so what we did for the study was, again, we're able to identify a gene fusion. So TP53 fusions are ones that we often see in osteosarcoma. USP6 fusions are ones that we often see in aneurysmal bone cysts. And this patient actually had a TP53 USP6 fusion where those 2 genes were coming together. And so this hasn't been reported before. And it's very interesting thinking about the tumor biology, which we don't have to get, too much into right now. But, this result was really unexpected. It was interesting in that it sort of explained, I think, a bit of the presentation that the patient had, which didn't quite fit with any aneurysmal bone cyst nor with an osteosarcoma. And then, using these results, and this was sort of in real time based on the histology, so what it looked like under the microscope, plus the knowledge of this gene fusion, the patient did get a diagnosis of osteosarcoma. and has been treated according to osteosarcoma protocols and is actually done very well. So that was an example of a patient who had a real-time impact on their diagnosis. And we actually had 17 patients, out of this group of 345 who had diagnostic changes. So they enrolled with this one diagnosis and then had either a clarification or a change in their diagnosis based on the molecular alterations that we saw. The other way that we can think about, diagnostic alterations in this group is that we can identify alterations that could have an impact on the, the patient's diagnosis. And there are a lot of different types of alterations that we expect to see, especially in a patient cohort as diverse as this. Excuse me, I'm just going to get a sip of water.
And while you're doing, so this is another like a beautiful graphic. And while you're getting that water, I might just set up the question, and you're probably going to do this anyway, but if you wouldn't mind just giving us like the brief primer on fusions versus sequence variants versus the copy number alteration.
Oh, sure. Yeah, this is a great time to do that because I'm going to talk about fusions again in a second. Let me get my water. So the gene fusions that we saw, like we saw in that prior patient where the TB53 and USP6 sort of each break off and then come together to make a hybrid gene, which leads to a hybrid protein. That's something that we're seeing over and over again here. But, especially if you look at an adult carcinoma model, we also see many sequence variants, and people often use the word mutation to describe just a single base pair change. So if we have like the DNA as like a long string and we have these A, T, G, and C in different sequences, and that's really, the foundation of all of the genetic code, you can have just a one base pair change that really, changes the whole protein and sort of the way that it works and can be like a strong driver. So in BRAF, which is a gene that we just saw in our other patient who had the BRAF fusion, we sometimes see BRAF point mutations, and those also can influence the tumor biology and could potentially be targetable. We also see copy number changes. This is like, now we're getting into the weeds of genetic, cancer genetics here, but that's another type of alteration that we can see in the data when we're looking through the next generation sequencing results. Did that make sense?
Yes, thanks.
So knowing that we have all these different types of alterations, because we just talked about fusions, sequence variants, and copy number alterations, if we look at all of the alterations that we saw in all of these 345 patients, and we ask like, how many of these could have an impact on the diagnosis or could sort of support the pathologist at the time they're making the diagnosis. And we can see, so in this pie chart, all of the 345 patients are represented there. And we've identified diagnostically significant alterations in more than half of these patients. And then we categorize them according to the diagnostic alteration type. So the fusions are shown in green, the sequence variants are shown in yellow, and the copy number alterations are shown in purple, and you can see there's a lot of green, right? So most of the diagnostic alterations are fusions. And then on the outer ring of this graph, we did another analysis to ask the question, would we have been able to identify these alterations using some of the older techniques? So we're really focused on using next generation sequencing, both the DNA panel and the RNA panels that we talked about. And so the pink is showing us ones that could have been partly identified, so the light pink, by using this sort of more old school techniques. The dark pink ones mean we wouldn't have identified these at all. And then that teal color means we could have identified them using some of the older technologies. And you can see there's not a lot of that teal because there's only 4 patients in that group where there was a diagnostically significant alteration that would have been completely identifiable using some of the older techniques.
And Alana, what is the white in the alterations by type?
Oh, so those are ones that didn't have. So not all of the patients had diagnostic alterations, but the ones that did. So those ones are the white, right? They didn't have an alteration that would have supported the diagnosis.
Got it. All right.
Yeah. So when we're putting all those together, we looked at another people graph where we saw how the molecular alterations could have an impact on their care. And then we're looking at this patient cohort and putting together, our 17 patients who had a clarification of their diagnosis, patients who had a recommendation for match targeted therapy, and either did or didn't receive match targeted therapy. And then we have that small group that had a significant response to match targeted therapy. So again, we can see that we were able to really influence the care of most of the patients within this cohort, which was a really positive outcome. So one of the other conclusions that we've sort of hinted at along the way is that the gene fusions that we're seeing here are really common and they're really important. So 27% of the therapeutic recommendations were made on the basis of fusions. Responder fusion of the diagnostically emission 70 fusion. And what's really important to make this point over and over again is that when we think about the molecular assays that are out there and that are available, if we're thinking about that adult cancer model where gene fusions are less common and less important, many of those assays don't actually evaluate for fusions, and they don't evaluate for the types of fusions that we would expect to see within this patient population. So we really want to advocate for including gene fusions as an important part of the molecular profiling process.
And Ashley, I'm just curious, because do most panels if they are adult panels, you're saying that they don't include many fusions? So I don't know, like, do places usually have like a separate panels for pediatric cancers, or is it usually just one panel? And
there's a lot of different models out there. So, I mean, for the OncoPanel assay, which was sort of the core technology that we were using for all of the patients in this study, we did, Katie and I and many others had influence over how that panel sort of came together from the design perspective. And we were advocating for the inclusion of fusions there. And it's not that people that are primarily focused on adult cancers don't care about fusions at all. It's just that it's a smaller proportion, and there isn't sort of as much data, especially on the diagnostic side, to sort of advocate for including those. And when you're doing a panel design, and this is something that I do and think about a lot, it's you can't put everything in, right? It's complicated. So you have to kind of prioritize and sort of focus on what is the most, going to have the most impact for all of your patients, and sort of look at all of those together. So the way that we have our molecular profiling set up now is that we do have different assays. So we have the OncoPanel assay, which has some fusions on it. And then we also are able to have our gene fusion sequencing assays in our lab at Children's Hospital that really focus on fusions. And so we evaluate a lot of fusions, and we wouldn't be able to fit all of those on the OncoPanel assay. There's just wouldn't be enough space. So that's the model that we have. Other groups do things differently. But there are a lot of new, really great technologies that really allow for evaluation of many fusions all at once, sort of in a single assay, which is great.
Yeah, I'm just thinking, because it's, I don't know if it's something I ever thought about as a caregiver, is kind of being that specific about thinking about which panel would be the best panel to use for testing. You kind of just go with whatever's available at your hospital, or like Foundation Medicine is, a third party that's used frequently. And I'm just wondering from like a patient perspective, or even the oncologist perspective, about like, if you have a pediatric cancer patient, maybe choosing that panel more carefully to make sure that it includes as many gene fusions as possible.
Yeah, I mean, I think that's a very, it is a tough thing to have to think about, right? If you're going through a new diagnosis and you have never thought about molecular sequencing panels before, many of the children's cancer centers, do evaluate gene fusion. This has changed a lot over just the last few years. So, we're not the only ones who have noticed that gene fusions are important, and a lot of the pediatric cancer centers and Foundation Medicine has a nice, list of gene fusions that are included in their assay as well. But I think it is a good point to sort of, begin to sort of ask those questions about gene fusions and making sure that everything's being tested, especially some of the ones that where we saw the responders. So we had like BRAF, ALK fusions, those ones that are really targetable and where there are drugs available now that work really well. You really wouldn't want to leave one of those behind. You know what I mean? If that was there, you would hate to miss one of those. So having covered those, I think, is a really important part of comprehensive care. And of course, we don't identify those in every patient. They're not there in every patient, but again, you wouldn't want to miss one. So this just making the point again, which you just sort of nicely led me into, is like, when we're looking at this workflow, what the NGS panel is actually matters, and sort of what tests are being done, how they're evaluating gene fusions, what list of genes are on the fusion, analysis really matters. So we're hoping to advocate for including gene fusion testing for all children with cancer. So our conclusions from all of that work and just looking ahead are that first, molecular tumor profiling has a significant impact on diagnosis and on treatment recommendations for young patients with solid tumors. These results really emphasize the importance of fusion detection for patients with sarcomas and other rare tumors. And we would like to advocate for the use of molecular profiling, and that it should become the standard of care on a clinical basis for children with cancer going forward. So there's so many people to thank, in addition to all of you, for being here with us today. The patients and families are so important, because all of this work really happened because they were willing to, share their tissue and to be a part of this study. We got generous funding support from several funders who are listed here. And then in addition to all of the many authors, so we had people from 16 different institutions that participated in the analysis of all this data. But we also had many physician and provider partners from all of these different cancer centers who referred patients and who supported the patients as they walked through the study. So thank you so much.
Thank you. That was, and I mean, I know that it's like so much work and so many years of work, and then kind of all represented in these like beautiful, concise graphics, which I know is a challenge to kind of put all that info into these, little graphics. So, but it made sense of it all. So thanks so much. Just a follow-up question on the gene fusions again and the different panels. I'm just curious, because there are, new drugs coming out all the time now. Like, is there any sort of kind of centralized management of saying like, oh, okay, like we know there's a targeted therapy for this, alteration mutation. Let's make sure that those are included in our panels. I mean, I don't know if like different institutions that have their different panels are just kind of doing that on their own, and that's just kind of pretty standard thing that people do, or but it would just be good to know that like, oh, okay, regardless of what panel we're using, that it's going to scoop up all the kind of, recent mutations where we know that there's like a targeted therapy available for that it could be actionable.
Yeah, I mean, that's a tricky question. And I will say that as like a molecular lab director myself, that, I'm very lucky to work with Katie and all these other wonderful oncologists who really spend a lot of time and energy in understanding what is available now and what might be available, because there's also a long fuse to like developing an assay and validating it and getting it up and running. So we don't want to only know what's available today. We certainly turn to Dr. Janeway and her colleagues and say what might be coming next year or the year after. And it is very complicated to keep track of all those things, and we sort of lean on one another's expertise. So the pathologists think a lot about the diagnostics. And so we kind of take the lead on that side of what are the alterations that might impact a diagnosis. And then the oncologists really, take the lead on what might influence targeted therapy. And we really have to work together. And it is it's an ongoing challenge, even as much as I spend all my time thinking about this, I don't have, I can't keep track of all of the drugs that are in there. And one of the supplemental figures that we had in the paper showed also the change. So for the same alteration, what sort of level of evidence it had at the time the report went out to the time when we did the reanalysis. And there was a ton of change. So things going in different directions, some went up, some went down, some stayed the same. And so there was a lot of work happening in the back end to keep track of all of that.
Thanks. And then I think we have a couple questions from the audience. Vicky, you wanted to ask those? Oh, and thanks. And I see Dr. Janeway's putting some more info in the chat.
Yes, great.
Yeah, I'm just going to come in with Ian Holsworth's question. He asked how many different tumor panels are available to children. He said Oncotype, Foundation 1, MSK, and are gene fusions currently being offered in any of the major profiling tests?
I'm not sure how many exact, I don't have an exact number, but I know that the children's cancer centers that I know of, the major children's cancer centers around the country, all have their own assays, and they are all aware of gene fusions and are, making efforts to evaluate gene fusions. Foundation certainly has, as we just said, like a very nice assay. They have a lot of fusions on there. Memorial Sloan Kettering has they see a lot of children with cancer there as well, even though they're not like that they're sort of a comprehensive cancer center, but they have a nice assay that's very similar to the one that we have in our lab in terms of the technology that's being used, and they evaluate a lot of different fusions at the same time. So these tests are out there. It's not that we're holding on to the one and only test that is available to do this. And I think a lot of the advocacy that we're hoping will come out of this is that any child with a new cancer diagnosis is getting access to one of these profiling tests, whether it's at our center or one of the other cancer centers or through one of the commercial labs.
Thank you. And the next question by Sabine was, how much time is there for testing, since a lot of osteosarcoma tumors can grow very quickly?
Well, that's a complicated question. It depends a lot on what's happening with the patient, right? So I think, where these treatments come in, and we don't have a lot of great targeted therapy treatment options that come up for most of our osteosarcoma patients, unfortunately at this time. That's why, sort of as Katie Janeway is pointing out in the chat here, it's so important for these drugs to be developed. And so that's a lot of what's coming out of like the Turbo and the Factor conferences, as some of these things going forward. Depending on what's happening with the patient, and again, this is like a conversation between them and their oncologist and their surgeon, but a lot of times at the early stages, the patients are following the standard of care. And I think there's a lot of good data to support that makes sense, for them to do that at that time.
And just a follow up to that. Oh, sorry, Vic, I was just going to pop in. I know Sabine asked a question, and it's kind of crossed over with the question I had too, which was, should testing be done before MAP is started, like at diagnosis, or is there still a first line, or is it still a first line treatment? And then kind of related to that other question about like in terms of recommendations for when you test, the importance of testing, as Sabine asked, like at diagnosis up front, and then the importance of testing over time. And do you see, because I think you did have a few samples in there where you kind of got some longitudinal data. Do you see the tumor profile changing over time, or does it remain pretty consistent with consistent drivers? And then I guess then the indication for like it's important to keep testing at each relapse every time you get a new sample.
That's a complicated question. I'm not sure that we have really data to make a kind of blanket recommendation, you know what I mean, about that. But we do see some changes that develop over time, for the same patient. And it depends so much on sort of what the genetic alterations were kind of at the beginning. And then you can develop kind of, clonal expansion of some, sort of populations of cancer cells kind of within the patient, also depending on the treatment, right? So some of the treatment, especially some of the harsh treatments can create sort of DNA changes as well, and that we might see. We haven't, yeah, that's a topic for future research. I mean, I think is to continue to look at that and to sort of see what we can glean from continued testing over time. And there's a lot of different things, you know, I, as a diagnostic person, would say it's great to do these right at the time of diagnosis, because if there was something that was going to influence the diagnosis, as we did see in some of the patients in this group, you of course want to do that earlier, right? You don't want to find out that you know, have been moving forward with the, you know, wrong information about the diagnosis until later on. So diagnostically, absolutely, you want to test as soon as possible. With respect to like the match targeted therapy treatment selection, it depends a lot on what that diagnosis was. So we want to make sure to get that right. But many of the patients, even who have targetable alteration, may not, you know, be eligible or want to pursue match targeted therapy early on. There was a few different things that you asked me there. Did I get everything?
Yeah, no, I think you did. And I also just wanted to point out, because, you know, Amanda Marinoff was on a few weeks ago talking about her work with MIC and MIC as a diagnostic biomarker. And so she kind of, you know, I think one of her points is that it's definitely like a great biomarker for risk stratification, perhaps even more so than what's commonly used today, which is necrosis, right? After neoadjuvant chemo. And so that combined, you know, work that like she's doing to do that combined with this. And what I really appreciate about this paper is that it was kind of making the case to say, hey, this should be covered by insurance, because this is clearly, you know, providing helpful data that is actionable. And so I'm just curious kind of how to, where we currently are to getting to the point where it does become standard of care that's covered by insurance. Kind of
the insurance reimbursement is such a complicated issue. And I would love to see some movement there where it becomes more standard of care. And it, you know, that was really a motivator for doing this study and sort of designing this interim analysis in the way that we did, to sort of get these data out there to show the benefit, right? To sort of generate some of that advocacy. So the insurance payers really all have different policies, you know, around reimbursement for molecular tumor profiling, you know, in adults and children. So there's a lot of variability in what people are getting access to. And, you know, again, we sort of use that early example in the early slides about lung cancer. And if we hope to follow that kind of model where we've seen that the data have come out over years about how important molecular profiling is for patients at the time of diagnosis, that it's really driving the treatment decisions, you know, both early on and as the patients progress, you know, if they have metastatic disease, for example, and need to change therapy, that a lot of those decisions are really made based on the molecular profile. And so that data has been really strong and has really pushed the field so that there are guidelines, you know, from professional societies that say that this is what we ought to be doing for every patient. And that kind of work, you know, really is what I think makes it difficult for a payer to say that, you know, that this is just a research study and that it's not really clinically relevant. And that is some of the feedback that we get sometimes, you know, when we've been trying to pursue reimbursement for these types of tests. This is a very complicated issue. And I think, you know, people who are listening who have some roles in advocating for patients, you know, can potentially have an influence there.
Well, this is another good, you know, like arrow kind of and the people can whip out this paper when they're kind of arguing with insurance on coverage. So thank you.
Yeah. Yeah, and I mean, the best thing to do if we can, and it's hard to do this, like I think as a you know what I mean, a person, you know, an individual, but is to really try to influence the policy, you know, not just arguing with a payer around a specific claim, but to try to influence some of the policy that, you know, sort of underlies the way that they kind of approach the claims that will be coming in going forward.
Yeah, definitely. Great. And I think
I'll just oh, sorry.
Oh no, I was going to say Vicky, I think, has another cue from the audience.
Yeah. Our next question is, of those molecular alterations that were tested in this study, what specific percentage of the alterations were druggable, and were there any alterations with a high prevalence that are not currently druggable?
There are a lot, I think Katie wrote a nice comment in the chat here about drugs and gene targets. So we have these big gene panels and we see a lot of alterations, and they don't all have, targeted treatments that we can even make recommendations for. So we do have, I don't know if I'm able to answer specifically what, there's a range of molecular alterations that we see, and we included all that data for this cohort, as part of the publicly available data set, to help researchers kind of going forward. And it would be great to have more drugs available to children to, TP53 was a great example that we saw in that graph, right? So we had a few years ago, there was a lot of recommendations were going out of the study for WEE1 inhibitors for patients with TP53 alterations, and we have many patients with TP53 alterations, whether they're gene fusions or mutations. And then that was one of the ones that went from being kind of a higher evidence for potential response to targeted treatment to being, the evidence kind of thinned out over the years. So if there was something that came in that would help there, that would be great. And we do have a large proportion of patients sort of within this cohort who had alterations there, and but there's not a great drug to recommend. So that would be a nice example. I mean, I think some of the other ones that we saw where we know there are drugs kind of out there are MEK inhibitors, PARP inhibitors, some of the other targeted pathways, and some things that are increasingly available for adults and maybe less so for children, but could certainly use some drug development in those pathways.
We have a couple more minutes, and Alana, I just wanted to ask this question too. So I think there were what, 64, 65 osteo cases, I think, yep, in the sample set. And then there were a couple of papers that were published recently by some teams at Memorial Sloan that they looked at similar to genomic profiling in soft tissue and bone sarcomas. And I think one of them looked at the data set using the MSK impact panel. And then one was actually looking at the Foundation Med database, and there were quite large numbers there, and but not pediatric focused. It was kind of all bone and soft tissue sarcomas. And I'm curious, at what point, how big of a sample do we need to get to a point where we can start to identify, and quantify the subtypes of osteo? So I know at Factor, actually, Dr. Janeway had mentioned, osteo is not, at one point, it's going to be different diseases, what kind of osteo do you have?
Yeah.
So wondering at what point can we get, because you have some data that were okay, this percent had a TP53, this percent had this type of fusion, and we have more data from other data sets. I don't know if it's comparing apples to oranges or not, but at what point can we then start to say okay, look, these are the different subtypes we're aware of, and this is roughly kind of how common they are, and how big each of these baskets are.
I don't have a number to give you, but I know this is part of the work that, you alluded to Amanda Marinoff's study that she did with Dr. Janeway. And, that was a great one. And we've seen this happen in other tumor types. If you think about neuroblastoma, which is another pediatric tumor, where the patients are really being stratified according to some molecular data, some other data like their age, their stage at presentation, and it takes a lot of work. And so that's part of why the work that Amanda did was so important, where you have that patient's outcome and their genetic alteration and all this sort of really granular clinical data about how they presented to create these molecular, partly informed by molecular, but groupings of patients. And it is challenging in any rare tumor to be able to get the numbers together to do that. So I don't know what the magic number is, but I think we're getting there. And it is sometimes hard to put data together from different centers, but the assays aren't so different that it's impossible to do that. And so, and pediatric oncologists in particular, I think, are very good at collaborating. So there are, quite a few collaborative efforts that are happening to put these data sets together. And then also, we spoke about this at the Factor conference too, but really making sure that people are doing the work as much as they can to share any data that you generate, right? Like we have the data from this study that, is now out there, and hopefully others can make use of it. And many of the other cancer centers are doing the same thing. We're also, happy to be working with the Count Me In study, and we'll be generating more data in that study with a real, focus on osteosarcoma, which will also be generating publicly available data sets. And so those efforts together, I think, will really support the kind of work that you're talking about creating these subtypes with a lot of evidence behind them to say why they're important. So it's not just that they look different under the microscope, but that to show that they really behave differently, and that they should be treated differently.
Great, thanks so much. Unfortunately, there was so much to talk about. We're already out of time. But we'll make sure, especially when we send out kind of a follow-up from this webinar, we'll include the link again to the actual paper so you can get all the details. Thank you so much, Dr. Church, for joining us on OsteoBytes today and for making it better for pediatric oncology patients everywhere. More information about this and all OsteoBytes can be found on YouTube and on our website at mivagents.org or your favorite podcast place. And next week, join us Thursday, July 14th. We're going to be talking to Dr. Chris Collier. He's a surgeon scientist who leads innovative basic science and clinical research to improve outcomes for his patients. And he's an assistant professor of orthopedic surgery at the Indiana University School of Medicine. And he's going to be discussing unanswered clinical questions in sarcoma, opportunities to move from bedside to bench with us next week. So thank you again to Dr. Church and Vicki for spending an hour with us today, and to our sponsor, the Osteosarcoma Institute. And thank you for joining us today on OsteoBytes. We hope to see you next week when we chat with Dr. Chris Collier. And in the meantime, you can go to our website at mivagents.org to sign up for Outrunning Osteosarcoma and to order a bow for Childhood Cancer Awareness Month in September. And also thank you to Dr. Janeway for being on and for helping answer questions in the chat and adding a little bit more color there too. So thanks everyone. Thank you, Dr. Church and Vicki, and we will see you next week.